curse is the disease. Dimensionality reduction is the treatment. Find the low-dimensional manifold where the data actually lives and work there. View projects the data onto the directions of maximum variance, the eigenvectors from the architect's toolkit. 20,000 genes collapse to 50 principal components. In this 50-dimensional space, distances are meaningful again. Nearby cells in PC space are biologically similar. FIN works. Clustering works. The curse is broken not by adding more data, but by recognizing that most dimensions are redundant. F finds a linear subspace, a flat sheet. But the manifold might be curved. UMAP and t-SNE find nonlinear structure by preserving local neighborhoods. A cell's nearest neighbors in 20,000 dimensional space are the same as its nearest neighbors in the two-dimensional UMAP embedding. UMAP is better than PCA for visualization because it captures curves. P is better for preprocessing because it's deterministic, fast, and the components are orthogonal. Standard workflow: pick a first to 50 dimensions, then UMAP to 2 for plotting. Instead of projecting all dimensions down, select the informative ones. Highly variable genes in single-cell analysis. Keep the 2,000 genes with the most variance across cells. Discard the 18,000 that barely change. Fewer dimensions means less curse. The Census Bureau pipeline selects HVGs PCA. This is feature selection, a direct attack on the curse. In high dimensions, every model overfits. A linear regression with 20,000 gene features and 100 samples has 20,000 parameters, 200 times more parameters than data points. The model can perfectly fit the training data and fail completely on new data. Regularization, the Leash video, is the antidote. Ridge shrinks coefficients towards 0. Lasso sets most to exactly 0, performing automatic feature selection. The curse demands regularization. Without it, high-dimensional models memorize noise. Hughes showed in 1968 that classification accuracy first increases as you add features, then decreases. The initial features are informative, they help the classifier. But eventually, each new feature adds more noise than signal. With a fixed sample size, there's an optimal number of features. Adding more hurts. In genomics, this means using all 20,000 genes is worse than using the best 2,000. Feature selection, HETA, and regularization all address the Hughes phenomenon. The curse of dimensionality is real. Distances converge. Volumes concentrate in corners. Thin breaks. Overfitting is guaranteed. But the manifold hypothesis saves biology. Cells live on a thin sheet in gene space. PCA finds the flat approximation. UMAP finds the curves. HVG selection removes noise dimensions. Regularization tames overfitting. The ballroom isn't empty. The guests are hiding on a thin sheet. Find the sheet and the party continues.